Okay, let's dive right into this explainer. Have you ever wondered how scientists actually manipulate something as unimaginably tiny as DNA? I mean, you can't even see it. Today, we're uncovering the real magic behind DNA extraction and genetic engineering. We're going to take what usually looks like dry, boring laboratory steps and transform them into a master recipe for molecular biology, pulling back the curtain on exactly how these invisible molecules are handled. Welcome to Molecular Mastery, the journey of a gene. In this explainer, we're decoding the exact methods, the techniques, and the reagents you absolutely need to know. Especially if you're gearing up for your CSIR Net Life Science exams, we're going to make sure these concepts actually stick.
So, here's our roadmap for today's lab workflow. We'll start with breaking in cell lysis, then move to cleaning up, nuclear purification, followed by cut and paste, cloning, and we'll wrap up with the molecular copy machine, PCR.
All right, part 1, breaking in cell lysis. Step 1 is honestly a lot like cracking an egg to get to the yolk. We have to break through the tough cell and nuclear membranes to release our precious nucleic acids without destroying them in the process. And this brings up our first major choice in the lab, physical force versus chemical precision. We could use physical methods, like ultrasonication. It uses high-frequency sound waves to literally burst cells open. Super fast, right? But the catch is, it generates heat that can actually damage the very DNA we're trying to save. On the flip side, we have chemical methods, which offer way more precision. Take lysozyme, for instance. Instead of brute force, it specifically hydrolyzes the bonds in the peptidoglycan layer of gram-positive bacteria, so it's more like using a molecular scalpel instead of a sledgehammer. Now, what's really fascinating here is EDTA. You're going to see this chemical everywhere in molecular biology. EDTA acts as your ultimate molecular bodyguard. See, once you burst a cell open, you aren't just releasing DNA, you're also unleashing nucleases, these destructive enzymes that just want to chew up your newly exposed DNA. But those nucleases need magnesium and calcium ions to do their job. EDTA actively chelates, or locks up those ions, completely starving the nucleases and keeping your DNA totally safe. Okay, moving on to part 2, cleaning up, nucleic purification. Now that we've burst the cell open and protected our target, we're left with this really messy cellular soup of proteins, lipids, carbohydrates, and our DNA. Time for the cleanup phase. So, how do we separate the treasure from the trash? We use phenol chloroform extraction, which relies on the fact that different biomolecules dissolve differently. When we add phenol, chloroform, and a bit of isoamyl alcohol, we spin it down in a centrifuge, and boom, we get 3 highly distinct liquid layers. The denatured proteins get trapped in that middle interface, and the lipids sink down to the lower organic layer. This lets us perfectly siphon off our pure DNA and RNA right from the top aqueous layer. It's exactly like skimming cream off the top of milk.
Time for a quick CSIR pop quiz to test your memory. Plant cells are notoriously tricky to work with because they're packed with polysaccharides and polyphenols that will completely wreck your downstream experiments like PCR. So, when extracting plant DNA, what specific reagent do we use to get rid of those pesky phenolic compounds?
If you guess PVP, you absolutely nailed it. Plant DNA extraction relies heavily on the CTAB buffer method. And while CTAB is busy clearing out the heavy polysaccharides, the PVP steps in specifically to yank out the phenolics. It's a real team effort in that little buffer tube to get pure plant DNA. Before we get to the really fun stuff, we have to check our work, right? We do this by measuring light absorbance. Molecules absorb light differently, specifically at 260 and 280 nanometers. A pristine batch of DNA will give us a very precise ratio of exactly 1.8. Now, if your number drifts way down towards 0.5 or 0.6, you instantly know you have dangerous protein contamination. All right, on to part 3, cut and paste, cloning. We have our clean, pure DNA, so now it's time to actually take that sequence and insert it into a vector. Think of this incredibly powerful 5-step process as molecular word processing. First, we cut the DNA with restriction digestion. Next, we glue it together into a vector. That's ligation. Then, we force that newly built vector into a living cell, usually bacteria, which is transformation. Finally, we grow the cells and use selection and screening to find the exact survivors carrying our specific edit. When we make that first cut, we use restriction enzymes, and they give us 2 totally different types of cuts. Enzymes like EcoRI create sticky ends. They leave these overhanging bases that easily snap back together with matching overhangs. Highly efficient, just like puzzle pieces locking together.
Alternatively, enzymes like SMAY-YI create blunt ends. These are flat cuts. They're way less efficient to glue together, but they offer universal compatibility because basically any blunt end can technically ligate to another blunt end.
But you really need to watch out for the supervillain of the cloning lab, a lab disaster known as star activity. This is a classic exam topic. It happens when your restriction enzyme loses its mind and starts chopping up incorrect, similar sequences instead of its exact target site. Why does this happen? Usually user error. Using way too much glycerol, having the wrong pH, or using excess enzyme makes your microscopic scissors go completely rogue.
Let's hit another classic CSIR exam question. Fast forward to the screening step, where we're looking at a petri dish full of bacterial colonies using a substrate called XGAL. Some are blue, some are white. Why do the successful recombinant colonies, the ones we actually want, show up as white instead of blue?
The answer is just brilliant molecular sabotage. Our cloning vector contains a reporter gene called LAC-Z, which normally processes XGAL to turn the cell blue. But when we successfully insert our foreign DNA, it physically breaks right through the middle of that LAC-Z gene. It totally disrupts it, meaning the cells literally can't turn blue anymore. So those white colonies, they're exactly the ones carrying our hidden genetic treasure.
And finally, part 4, the molecular copy machine, PCR. This is the technique that lets us take a tiny, practically invisible amount of our cloned DNA and amplify it into millions or even billions of copies. PCR is basically just a game of extreme temperature cycling. First, we heat the reaction to a staggering 94 to 95 degrees Celsius. This literally boils, well, actually melts apart, the double-stranded DNA into single strands. Then, we quickly cool it down to around 50 to 65 degrees so our custom primers can find their target and attach, or anneal. Finally, we warm it back up to a perfect 72 degrees so the polymerase enzyme can race down the strand and build the brand new DNA extension.
Now, not all of these molecular copy machines are created equal. Taq polymerase is the industry standard because it survives all that extreme heat, but it's actually pretty sloppy. It has no proofreading activity, which means low fidelity. If you need a perfect, error-free copy for cloning, you want to use enzymes like PFU or PFX. They possess active proofreading abilities so they can actually catch, cut out, and fix their own microscopic typos.
To make PCR work without a hitch, you've got to design perfect primers. Keep them around 18 to 25 base pairs long, with about 40 to 60% GC content. You absolutely should utilize a GC clamp, meaning 1 to 3 G or C bases at the 3 end, to securely lock down the primer to the template. And whatever you do, actively avoid sequences that cause primer dimers, where your primers accidentally just bind to each other instead of your DNA.
Let's do one final rapid-fire check to make sure you are totally exam ready. We've talked a lot about DNA, but RNA is notoriously fragile because RNase enzymes are practically everywhere and super resilient. What chemical reagent is absolute kryptonite to RNases, keeping them completely inhibited during RNA isolation?
That's right, it's guanidinium thiocyanate. This stuff acts as the ultimate protein terminator. It's a super powerful tropic agent used in trazol extraction, and it utterly destroys RNases on contact. That way, your incredibly fragile RNase stays completely intact for your downstream work.
Everything we've discussed really culminates in this simple equation, which represents the terrifying and beautiful exponential power of molecular biology. By extracting just one pure template and cycling it through PCR, you are literally turning a single microscopic strand into over 1 billion copies in just a couple of hours.
So, as you master these foundational lab recipes, ask yourself something. If we can already extract, cut, and infinitely copy the fundamental code of life in a simple plastic tube, what incredible new genetic techniques is your generation going to invent? What biological mysteries are still hiding just beyond our current toolkit, waiting for you to decode them? Keep learning, keep studying, and we'll see you in the next explainer.